The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Now, before we get started, I'd like to introduce our speaker. Dr. Judy Van de Water's laboratory pursues research programs pertaining to autoimmune and clinical immune-based disorders, including the biological aspects of autism spectrum disorders. She's currently the director of the NIEHS-funded Center for Children's Environmental Health at UC Davis, investigating potential environmental risk factors contributing to the incidence and severity of childhood autism. In addition, Dr. Van de Water's work is also part of a comprehensive and multidisciplinary analysis known as the Autism Phenome Project. These webinars are made possible through generous donor support. If you'd like to contribute, please visit our website at autism.com. And now, if you have any questions now or throughout the webinar, be sure to type them in the question section. We'll answer as many as possible at the end. And I will turn this over to Dr. Van de Water.
Great. Thanks, Denise. And thanks, everybody who's joining today. Hopefully this will be both interesting and informative. And please, I welcome any questions as I'm presenting. If there's anything you don't understand or anything that you'd like to know more about, please let me know. So today, what I'm going to talk about is a large project that is ongoing in my lab, which are the immune issues in autism, the gestational profiling, what's going on during pregnancy that affects neurodevelopment. And so the overarching goals of our research at UC Davis really, as Denise said, I'm the director of the Center for Children's Environmental Health, so the environment is a big part of what we do here. And we're looking at the relationship between the environmental factors, genes, genetics, how, susceptibility factors from genetics play a role, and the immune system as they relate to autism susceptibility. And a large part of what we look at is the gestational immune environment in autism. And so how changes in signaling molecules such as cytokines and chemokines that I'll explain a little bit more about later affect neurodevelopment. And this is what we call sort of the infection model or the maternal immune activation model, what happens when the maternal immune system is activated during pregnancy. And then the second part of what we look at is the maternal autoantibodies that mothers make that recognize proteins in the developing brain, and that those antibodies can find their targets and have an effect on changing how the brain develops. And this is a percentage of women who have children with autism have these autoantibodies, and that is a large part of what we're doing, and so that will be the second part of my presentation. And just sort of to remind everybody of, while we all know the core autism spectrum disorder symptoms of impaired social and verbal communication and restricted repetitive behaviors, as is the new DSM-5 criteria, we also recognize that there are a number of other sort of, I guess we call them comorbidities, but other issues that arise in many children with autism. And we also recognize that this is a very broad umbrella under which there are many levels of affectedness, but those include anxiety, language impairment, intellectual disability. We know that there are some genetic single gene mutations that can have autism as part of that, but also we know there are a number of medical comorbidities, including GI issues and seizure disorder, but in the area that I'm working in is the biomarkers. And so this as I said, there are many other health issues found, and looking for biomarkers and treatment in both the prenatal and postnatal period have become a strong area of interest. And then my area would be what's going on in the immune system of both the children with autism and mothers of children with autism, so that we hopefully at some point can come up with preventative or therapeutic interventions. And so our conceptual model really is, as I said, the environment, I'm sorry, my mouse is very sensitive to my fingers there, is the environmental factors as they interact with sort of the immune, endocrine, metabolic, and even the role they play in epigenetic changes of our genome. Genetic factors is an underlying susceptibility where a gene may not cause a disorder, that gene could make you more susceptible given an environmental input to having a disorder. And then how environment directly affects development, because we know there are some environmental toxic exposures that can directly affect how the brain develops in neurons if it's in the system, and the genetic factors that affect the medical background upon which we may have susceptibility to having autism. So this is really how we look at that at autism here at Davis. We don't look at a single entity on its own, but we really try to understand the relationship between all these entities. So I think key to understanding what I'm going to talk about as I go through this presentation is understanding the immune system. And to really do that, you have to be familiar with how it works. So I'm just going to give you a quick primer, and this slide is meant to be as complicated. This is what we deal with. This is the complicated immune system that we try to tease apart and find those areas that might be contributing to changing neurodevelopment. And as you can see, it's a very complicated. There's a lot of jargon that we use as immunologists, which I apologize for. But I'll try to break this down into a little simpler basis for understanding. So it's a complex, as you saw, and very developed collection of both cells and tissues and molecules that their whole job is to resist or prevent and cure infection. And there's a fine line that we have between having a very active immune system that can actually protect you, but also one that doesn't overstep and then cause problems on its own, which is what we call autoimmunity. So to prevent and eradicate established infections and distinguish self from non-self, which is the last part I was talking about, because the immune system can turn its artillery on our own bodies, which is what we call autoimmunity, there are checks and balances in place to prevent that. And sometimes those get overridden, and we end up with an autoimmune disorder or autoantibodies, cells that recognize our own tissues. And so that's what the immune system is trying to balance to keep us healthy. The innate immune system is what we call, this is your first line defense. It's critical to the adaptive immune response, which is what your sort of the more specific response to a pathogen. But the innate immune system is what comes into play for every infection, and it's got receptors on its surface that recognize elements of a pathogen or a bacteria or a virus that then arms that cell to both fight that pathogen, but also to signal to the adaptive immune system that we need to increase the army against this particular pathogen. And that would include the effector T cells, or these T cells that are directly fighting the pathogen, but also the T cells that help, that tell cells to make antibodies. And that comes into play when you, especially for certain long-lasting immunity, that those antibodies are in your system. So where the nervous system intersects with the immune system, we now know that they do interact, and it was for a very long time, it was thought that these were completely separate systems and that they had nothing to do with each other. And as I mentioned, the immune system has a broad set of receptors and cells to recognize a wide variety of foreign agents. The nervous system transmits signals, and it is also a very complex interactive system with signaling molecules of its own. Where these two come together is where we do most of our work now. And the complex interaction happens both during health. The homeostasis is a good thing. It's actually what provides you with memory. The immune cells that are in your brain at all times help you maintain your memory throughout your life. And in response to injury, it's there to protect you and to repair. Some of the cytokines that we think of as inflammatory cytokines actually are doing a job when they need to help repair the system. But it's also, and this is where my work focuses, particularly important during development. And so the elements, the key factors that we look at in those terms are the cytokines and chemokines. So those are the signaling molecules that tell immune cells what to do. But they also talk to the nervous system, as I'll show you in a graphic in a moment. But also antibodies, and particularly autoantibodies. So cytokines get a lot of people talking about inflammation, and then when they're referring to inflammation, this is usually what they're referring to, or the cytokines and chemokines. And really what cytokines do is they are small molecules that talk from cell to cell and tell this cell to quiet down if they're a regulatory cytokine, or this cell to ramp up because we're being invaded and you need to divide, we need more of you, and we need you to do this job. And that's really what they do. And a lot of different kinds of cells can produce cytokines. They direct the function of the cell that produces them and the cells nearby. They're produced by a lot of different cell types, including T cells, B cells, and macrophages, but also sometimes even fibroblasts and neurons, as you'll see in a minute. So as I said, cells in the central nervous system do produce cytokines and chemokines, including microglia and neurons. Pardon me, I'm getting over a cold. And so in this graphic, really the point I'm trying to make is that while we know these are 3 inflammatory cytokines, IL-1 beta, TNF-alpha, and IL-6, that are involved in the pro-inflammatory pathways to fight infection, they also have a very important role in the homeostasis of sleep and your fever response. This is the response, these are the cytokines responsible when you have a fever for that lethargy you feel, that you want to just sleep, and also increasing your temperature so that you make your body inhospitable as a host for that whatever the infectious agent is. Unfortunately, the IL-1 beta in particular can also be neurotoxic if it's a sustained high dose of this cytokine. And so where TGF-beta comes into play, this is our anti-inflammatory cytokine, along with IL-10, that downregulates this response and is involved in almost all aspects of neural development. And then we have the chemokines, and these are just some examples of the chemokines that are involved in getting cells where they're supposed to go and to the site of infection, but also very important during neurodevelopment for oligodendrocyte function, interneuron migration, especially when we have microglia migrating in to do pruning during brain development, and neuromodulation. So this is an old graphic of immune crosstalk. So that used to be the blood-brain barrier was thought of as a barrier that didn't allow anything across. There was no talk between the periphery, or the blood system, and the brain, and that neurons didn't produce cytokines, they didn't have receptors for cytokines, and that immune cells didn't have neurotransmitter receptors on their surface, and they didn't produce neurotransmitters. Well, we now know that this is not true. We now know that both systems contain both of these, both immune and neurotransmitter receptors and the production. So this probably in the last 5 years has been, 7 years maybe now, has been the biggest change that we've seen, is that this crosstalk is important. Cytokines get across the blood-brain barrier because they're very small molecules. They are transmitted into the brain. Cells only get across when something is upregulated on the blood-brain barrier and tells the cells that we need you in here to do a job. Antibodies are very large and don't cross unless there's truly been a large breach, but some are made locally, actually, in the choroid plexus of the brain. So what is our evidence of immune dysfunction in autism? Well, we know now that the genes that they have shown in as being autism-specific genes that they have found that are upregulated, say, in brains, the expression of them is upregulated in brains of individuals with autism. Many of those regulate the immune response. Many of those are responsible for what we see in the genetic differences between a typically developing brain and a brain from an individual with autism. Abnormal immune markers in the peripheral blood of children with ASD. We see altered cytokine profiles, and that's another subject of what we do here. But we do see changes. This is less mature at this point because I think there's been a lot of discussion that inflammation is key in autism, and I think that is not accurate. I think there are some children for whom the immune system is not regulated well, and they do have a lot of inflammation. There are other kids where I see their immune systems barely function. So we need to be very careful, but that's a topic for another webinar. Neuroglial activation and neuroinflammation in the brain and the CSF has been shown by some investigators in brains, archived brains of individuals with autism. So this work is very important, but that's just, it's a snapshot in time, right? So we are working toward understanding that a little better now with our animal models. And then infection associated, increased number of infections, asthma, allergies in children with autism. Many kids, a lot of the population has asthma and allergies, and they don't all have autism, but I think it's particularly important when we look at the immune system in children with autism to see where, because that is considered sort of an immune dysfunction because you're hyper responding to your environment, but also the downstream effect of having allergies and asthma in children with autism can certainly affect their behavior. It affects all of our behavior, but they're especially going to be sensitive to that. So the maternal immune environment really is going to be the focus of the rest of this presentation. And this, really the question we ask is, does changing the immune profile of a mother during pregnancy alter development in the brain of the fetus? So the evidence for an association with autism has also come from work looking at infection in the mother, asthma, allergy around the time of gestation, autoimmune disease during pregnancy, maternal history of autoimmune disease or familial history, autoantibodies that we found here in the serum or plasma that recognize fetal brain proteins, and I'll talk a lot more about that, as well as altered patterns of inflammatory markers in either the serum or amniotic fluid, and this includes the cytokines and chemokines. There are several unanswered questions, however, because the maternal infection in ASD has gotten a lot of press. This is what we call the MIA model, maternal immune activation model, or infection. And I think we need to be very careful how we think about that because, first of all, a lot of us get sick when we're pregnant because your immune system is downregulated, because you don't want to reject the fetus. So that makes you a little more susceptible. And as we've all been pregnant, we've all had, you get worse, the colds and you can't take anything for it. And that's not what we're talking about here. And I'll explain more about that in a minute. But the other thing is, what agent? Is it a specific agent? Is it viral, bacterial, parasitic? What is the timing? What's the importance of timing during the first, second, or third trimester? Because the brain is at different points of development, as is the immune system of the fetus and the mother is in a different state during each of those trimesters. Is it the agent itself, or is it really the immune response to the agent that is causing this? Fever, is fever required? And it's looking like fever is certainly one aspect, that you get to the level of infection that you mount a fever. But keeping in mind that how long is that fever? How sustained was it? How high is it? Is your response get dampened down in a timely manner? Increased production of cytokines directly or indirectly impacting the fetal brain. Is that, how is that working? Is it working through the placenta, directly on the fetal brain, both? And these are what we use our animal models to understand, and I'm sorry to say that we aren't at the point of completely understanding that yet. And then the underlying genetic susceptibility, and this is an important point. While we don't understand all of the genes that are involved here, we do know that genetics always underlie everything, right, that controls our system, but a deficiency or something going on in a certain gene may make you more susceptible because maybe you're lacking control of the regulatory system isn't in play as much as it should be. So those are the other things that we're looking at. And there's a number of papers, and I'm just going through this as an example. So even going back with Stella Chess's first paper on congenital rubella and autism back in 1971, and just through time more and more has come up, and so I think it brings to mind that this is a place we need to go. So all these epidemiology studies help us, point us in the right direction. And we, like from this particular paper that was done here at Davis, was it influenza or actually fever? And it turns out it was fever that was the issue, not influenza itself. So we need to be careful. It's not to say a certain infectious agent, if you get the flu, you're going to have a child with autism. No, that is not what we're trying to say. It's just for some individuals, having a fever during pregnancy that doesn't resolve either quickly enough or gets to a very high level, or perhaps even overlaid on a genetic susceptibility in the child, can potentially change neurodevelopment. So these are just a number of papers that have come out from a number of investigators, which just strengthens the idea that this is an area that needs further study. Most studies show, so the maternal immune-mediated disorders and autism, many studies show an association, but the specific infections and the specific autoimmune diseases vary, right? And so timing during pregnancy varies. That is also a critical component. Do you end up with schizophrenia or autism depending on timing? These are all the studies that are ongoing here at Davis and in other institutions. Increased production of cytokines directly or indirectly impacting, as I said, the brain. Autoantibodies, how do those come into play? How are they generated? And that is a question we may never have the answer to. And then what is that underlying genetic susceptibility? So this is some work that I did with Dr. Lisa Crowan at Kaiser, and what we found was that if diagnosed within 2 years of pregnancy, either before or after pregnancy, psoriasis and type 1 diabetes were the two autoimmune disorders that were most frequently associated with having a child with autism, with risk. The asthma was also highly associated, as was any allergy, especially atopic eczema and allergic rhinitis, which is hay fever. So while this is not definitive, and as I said, it's not every case, it helps us understand what might be going on. And the period in which this seemed to be most critical in terms of asthma and allergies was the second trimester. That is the period where your immune system during pregnancy is the most suppressed. That's the period where it's most critical that it's suppressed. So one of the things that we do is we can measure maternal cytokines mid-gestation to better understand which of them, what the profiles look like, even and what phenotype of children, of kids on the spectrum might associate with that. So we did, I'm part of the early markers for autism study with Lisa Crowan at Kaiser, and this was a case control study, prospective study, meaning that samples were taken from banked plasma that was taken for alpha-fetoprotein testing mid-gestation. And that's what we used to look at that. So we studied mothers of children with autism, mothers of children from the general population. The caveat of this study was they weren't necessarily typically developing because we didn't assess them for their development. They just were families that had never sought services for that child. So they could have had some underlying issue. So it's not the cleanest control population, but it was a very large sample set, so that helps with that. We also had mothers of children with developmental delay without autism. And this is an important control group for us because there is intellectual disability and developmental delay in some children with autism, and so this is a control because it's lacking the other phenotypic characteristics that gives you a diagnosis of autism. So the two things that we're looking for were early biologic markers, so that's the area that I'm working in, and then determining the contribution from the immune system, genetic factors, as well as the environmental exposures and how they interplay with each other. And this is from the first, so we had two phases of EMA. Phase 1 was a very small study, and from that study we found when we measured serum cytokines, so we measured several cytokines, the three that showed the biggest difference were interferon gamma, IL-4, and IL-5 in between the mothers who had children with autism versus mothers of the general population controls and mothers of children with developmental delay. Now, keep in mind there's a lot of overlap. It just means that more frequently we saw higher levels. It wasn't a complete separation where if you had high interferon gamma, IL-4, and IL-5, you had a child with autism. It just, that happened more frequently. And interestingly, IL-6 was actually more associated with developmental delay without autism and separated from both populations. So this is contrary to sort of what the current thinking is from the maternal immune activation model, which is really driven by thinking that IL-6 is the important cytokine. So what does this tell us? So just from this part, we know that increased interferon gamma, IL-4, and IL-5 are consistent with an allergy and asthma phenotype. Those are, interferon gamma especially is what we see elevated in asthma, IL-4 and IL-5 in allergic rhinitis. Placenta forms a barrier between the maternal and fetal circulation through maternal immune factors, including IgG and IL-6. IgG and IL-6 are permitted to cross, but we don't know what of the other cytokines and chemokines are. So even if direct passage is blocked, however, maternal immune components could react at the placenta and could then change what's going on in the fetal compartment. And so these are all studies that we're trying to understand. And this may be the case for interferon gamma, IL-4, and IL-5. And my mouse just decided to disconnect, so I have no pointer at the moment. But mothers of children with ASD, in the second phase, we had a much larger sample set, which allowed us to subphenotype children who had ASD with intellectual disability and separate those from children that did not have intellectual disability. And this increased our power greatly, and this is a paper that my postdoc Karen Jones put out recently in molecular psychiatry. But what we noticed was, if you look at where you see the red, between all of autism and the general population controls, or DD, we didn't really see a significant difference for these cytokines. But when we broke them down into with intellectual disability and without intellectual disability, we saw that mothers of children with ASD plus ID had elevated inflammatory T cell and innate immune cell cytokines compared to every other group that we tested them against, including mothers who had children with ASD without intellectual disability. So these are all supposed to be downregulated during gestation, as I said, and it suggests a lack of typical immune regulation during pregnancy. And for some, I'm going to try and turn my mouse back on here. But and so this was a really great clue for us that, first of all, autism with intellectual disability is likely arising differently than developmental delay or intellectual disability without autism. And I think that was a huge thing for us. And also that we could just phenotypically, based on this, distinguish between two populations within autism was also very important. And so we will try to continue this work and model that further in our animal models. So moving on from that, and so that's sort of where we are here in the state of what's going on, and the animal models are also working along these same lines. But I'm going to move on to the maternal autoantibody related, or MAR autism, and this is where it's just to back up a little bit. Autoantibodies are antibodies that are made against our own proteins. We don't know why it happens. We think it's issues with immune regulation, but this is just to let us know that autoantibodies do exist, and even though the immune system is supposed to regulate and prevent them. And then antibodies in MAR autism are present during pregnancy and recognize proteins in the developing brain, and this is an important part for the rest of this presentation as to what's going on. And so what this is, the model is that some women develop, we don't know why, antibodies to their own proteins. We don't even know that pregnancy is important for the development of these antibodies. I suspect that it's not in all cases. These antibodies cross the placenta, and IgG crosses the placenta to protect your fetus from everything that you've been exposed to. It's our way of passively protecting the fetus as it grows, because their own immune system doesn't really mature until after birth. And so, as a matter of fact, antibodies stay in the system for at least 9 months postnatally. So they're a passive protection, and it's not in all mammals, but in most mammals this is the case. Now, unfortunately, if there are antibodies in the system that recognize proteins, self-proteins, such as heart or neuro tissue, brain tissue, they will also recognize and bind to those, and that is where the work that we're doing comes into play. And the result in that could be altered neurodevelopment. So as we, I'm just going to grab my other mouse so I can actually have a pointer here. So that is what we're working on in terms of trying to understand how that's happening and create models to better understand which system we're impacting. But what we do know in children with autism whose mothers have these antibodies have more severe behaviors, pronounced especially stereotypic behavior, and significantly larger brain volume, especially in the boys. So I will show you some data on that. So there's a lot of work has gone into this, and initially it began with this blot up here where we see the upper and lower band. We took fetal monkey brain, we ran it on a gel to separate the proteins by size. transferred that to a membrane and probed that membrane with plasma from mothers who had children with autism, who had children that were typically developing, and who had children with developmental delay. And when we saw this pair of bands, it was only in the autism, and that was really the genesis of a lot of the work that is going on now. As I said, these are associated with, we saw early on it was associated with regression, and that still is true, it's just not as high as it was when we did more of them, but we do see more often with regression. Language deficits and stereotypic behavior also with that particular pattern were highly associated. In genetics, we did find that CMET, which was found by Dan Campbell and Pat Levitt when they were at Vanderbilt, is a variant that they've associated with in children with autism. In the mothers, we found that this variant was associated with the production of these antibodies because MET in the immune system as a receptor is a downregulator of immune function. So when it's not functioning correctly, you have sustained inflammation. So you have once you have a response, it doesn't cool down as quickly as it should. When we've done MRI, we've seen enlarged brain volume in male children prenatally exposed to these antibodies. We also have now figured out what the targets are. There are actually 8 targets now that we know of. And it's a combination of reactivity of those targets that gives us our biomarker. And then we have several animal models. We've now got 2 monkey and 3 mouse models that show behavior changes when they're exposed prenatally during gestation to these autoantibodies, and I'll talk more about those as we go through. The monkey and mouse models also reported increased brain volume. So what we see in the humans is translating to our model, which is great for us because that's how we will be able to answer, hopefully, some of these questions. And then the brain tissue studies are ongoing to understand exactly what cells are being affected in the brain. The translational potential for this is that we could identify kids with this subphenotype and hopefully develop more tailored behavioral treatments for them, because if we can figure out what works in those kids, we can go right to that therapy. And that's, if we have a biomarker for those kids ahead of time, we know exactly what we need to do. And so we're working on that here at Davis. And then we can screen women at risk and hopefully in the future develop preventative strategies. That's, of course, our ultimate goal, is to be able to prevent this more severe form of autism in the future. And then if we can define the pathophysiology, meaning, what pathways are affected, what parts of the brain are associated, perhaps we could actually develop therapeutic interventions that would be effective postnatally to fix those pathways. So there's a lot of evidence now that this is truly something that is specific for autism. A number of papers by both our group and others, Harvey Singer and Andy Zimmerman, when Andy was at Johns Hopkins, had done the work in this area. Brimberg et al. At the Feinstein in New York are also working on this. Each of us have approached it differently, but we've all come to the same conclusion that women do harbor, some women who have children with autism have autoantibodies that then affect behavior or long term. So there's a number of animal models as well showing that if you transfer the antibodies into the gestating female animal, that the offspring have the offspring are affected. And so, but I'm going to show you a little bit more about that in a moment. And as I said, the autism is more severe with these antibodies, and this was especially true in the boys versus the girls. And so why that is, I don't know, because in other aspects there doesn't seem to be a gender difference with these. We saw, as I said, we have abnormal brain enlargement in children with MAR-ASD when we did MRI. And so I'm sure many of you are familiar with studies that have shown enlarged brain in some children with ASD. And you can see there's quite a range here from very large to actually small. But these are statistically significantly more enlarged than those brains at the same age-matched brains of typically developing children. This group in the middle are boys that came from mothers who had the autoantibodies, and it's a very tight and significantly higher than either population in brain site total brain volume. So I think what we're seeing is a physiologic effect of these antibodies, and it's starting to explain some of the other findings in autism. this is really not going to mean a lot to anybody except just to show you that we know what the targets are. Actually, there's an 8th one that I need to add in here when we get through our intellectual property issues with it. But each one of these proteins that these antibodies recognize, and so STIP1, CRIMP1, and LDH were the first 3 that corresponded to this pattern. That was our main pattern. That's the one we see the most. But each of them has a different role, from neurodegenesis, dendritic branching, on to axon outgrowth and growth cone collapse in the development of a neuron. So they each have a role. And so understanding that and knowing that you need a combination of them to really fully impact neurodevelopment, it's an additive effect is what we're seeing. Because alone, one of these antibodies is not enough to fully impact the system. And part of that is because, first of all, the antibodies don't start crossing the placenta until day 100 of gestation. So any earlier than that, they're not going to have an effect. And just to put it in perspective, the human brain consists of it's almost about 100 billion neurons. And during the last trimester of development, they form at a rate of 580,000 neurons per minute. So you can imagine that there are always more proteins present in the brain from these developing neurons than there are antibodies, autoantibodies. They're going to be there. So we won't hit every single cell, and we won't, because otherwise we would probably really change, and you might not have brain development would be even more impacted than it is. So what are the effects of these antibodies? Do they have pathologic significance? And another way of saying that is, do they cause disease? And because they could be an excellent biomarker and say, yes, if these are present, you're going to have a child with autism. But we need to prove that they actually, because if we want to go to a therapeutic avenue, we need to prove that they have, that they cause pathology in the brain. So one of the reasons for this is we know that human behaviors corresponded to the autoantibody pattern, and that we saw lower expressive language and increased stereotypic behavior, especially with the LDH, CRIMP1, STIP1 pattern. So we took advantage of that, and we transferred IgG from humans that we purified that had reactivity to these 3 proteins into pregnant monkeys, and they got 6 injections beginning at the first trimester, so that they were on board when the antibodies started to cross the placenta in these monkeys. And this is work that I did in conjunction with Melissa Bauman, my colleague here at the M.I.N.D. And so we looked for the effects both physiologically and behaviorally. And in the behavior, it was really interesting that the mothers of the MAR IgG treated monkeys, so the ones who had the pathologic antibodies, seemed to be overprotective of their offspring. And it was as if they recognized that there was something potentially wrong in these animals, that they weren't interacting normally, but it also, by doing that, they're going to change their socialization. So that was sort of a 2-pronged issue there. They weren't going to learn to socialize correctly if they weren't socializing, if they weren't allowed to socialize as much. As they got older, they definitely showed evidence of abnormal social behavior with both familiar and unfamiliar peers. They would go up and approach and try to interact with their peers, but whatever they were projecting was not received in a typical way, and that so that the receptive animal, so that the ones they were approaching, did not engage in species-typical social behavior back. So they weren't projecting typical behavior to their peers, and therefore the reception was not typical as well. And we were fortunate in this study that we could image them over time, and we could do MRI imaging of the offspring over time. And at the same, we saw an elevated growth trajectory of the brains, especially in the male monkeys, which mirrored what we see in the children with autism. And so just to remind you that we're seeing a similar pattern. And then in another study, because mice are much cheaper and easier to work with here, we looked with my colleague Veronica Martinez-Sardeno here at Davis, also looked for prenatal exposure to autism-specific maternal antibodies, and we found just by 1 injection she injects them, we purify them and label them, and she injects them into the ventricle of the brain. She actually goes in while the fetus is developing and with a very fine needle can inject right into the ventricle of the brain. And this allows, she was using this technology for something else, and I co-opted her into helping us with this. And then we analyze, she can put them back in and they develop perfectly fine. They're that good at this. And so for some, we actually labeled the antibody before it went in and looked at what happened to that labeled antibody, and that's what it looks like. So the antibodies were injected here. We put a label on the antibody that was injected in, and then when she took the brains out 2 hours later, she could stain them for that label. And this is when we take typically developing the IgG from a mother who had typically developing child and injected exactly the same way, we get no staining. So the more brown there is, the more antibody there is at the site. And so the interesting thing from my perspective is that these antibodies are actually getting into the cells. And what they're getting into is at this part of the brain right here is where the stem cells are producing the new neurons. And they are taking up these antibodies, and it's changing the way they move through the cortex. And so what they're doing is changing, and you can see a nice picture here. This is a control, and I thought we'd screwed up. I thought, oh, we didn't get the label on it, and when we went back and redid it, we had plenty of label on there. It just doesn't stay long enough for us to be able to pick it up and stain as the antibodies are finding their target and staying inside. So you can see all through these the dendrites of these are stained. So the conclusions from everything I've presented really distill this down into the sort of the big picture here, is that we are really working to understand how changes in the mother's immune system during pregnancy contributes to the risk of autism. And we think this may be underlying the mechanism behind increased risk due to infections during pregnancy, and it's really the lack of immune regulation in the mother that is driving this. And then the mouse model that I just presented, so my most recent model, is the only ASD model actually out there right now that captures all three domains of autism with the social, verbal, and stereotypic behavior deficits that are strongly reminiscent. And the clinicians that watched those videos actually were astounded. It's like watching a child come into the waiting room, with a child with autism. So it's exciting because now we have a model, an animal that we can both study and to understand what's going on, but also that we can start to work on a therapeutic intervention for that using that model. And then I think we're confident that autoantibodies, they change how the brain develops, and that they are a good biomarker, and that we can hopefully develop this into something that will be useful in predicting so that we can have treatment options available. So I think why this work is important is that we hope to find patterns of maternal health conditions and biomarkers that indicate risk, because hopefully some of them we can prevent, and that we can come up with strategies to lessen their impact. And early identification can lead to earlier intervention, and I know we talk about that all the time, but not just earlier, but more effective. Finding the right intervention that works on kids, that come from a particular cause. And then the identification of biomarkers for prenatal risk will hopefully shed light on biologic mechanisms underlying changes in neurodevelopment, providing an opportunity for developing preventative strategies in the future. And I don't do this myself. Karen Jones, who's sitting right back here, is the person who does all of the animal behavior that we do here, all the mouse behavior, and she's key for that. And so I have a great team that supports all this work, and my collaborators. And so if there are any questions, hopefully I've left us enough time for some questions.
There's several threads in the questions, and I think one of the big questions that came up relates to the recent study that I'm sure you've been following about stem cell research, and how that relates to what you were talking about with the mouse models, and also with the staining that you described. So do you see ties between the research you're doing here directly to those recent findings that were announced last week, or where do those two things dovetail?
And that is a great question. So the stem cells I'm talking about are the ones that are already at the ventricle layer of the brain. They're not bone marrow stem cells, which is what they're using for that therapeutic. Just to distinguish those two, we use stem cell as it's a generic word that we use for cells that are progenitor cells that turn into something else, and they can come. They're in many places in the body. So, I think the hope for that work is that you're providing stem cells that can then, with the right signals, generate new cellular tissue. And I actually, Jerry Dawson asked early on if I would be part of that, and I think there's trying to understand what the immune system looks like in the kids who are going through that. So they've saved samples for when we have funding to do that, to try to see what the overlap is, if there is any overlap. So I'm hopeful that in the future we'll be able to participate in that, and because we don't know what the connection is right now on that. Hopefully, did that answer the question? I hope.
Yeah, I think distinguishing between the different stem cells is important for everybody who's listening. The next question is about postnatal viral exposure and other environmental insults that can happen postnatally. How that relates to the antibodies that you described. So if a child had those exposures while they were gestating, and then after they're born, they acquire a virus or they're exposed to something in the environment. Has there been any research done on that higher susceptibility that directly points to a connection?
I think most of the work, not for the autoantibodies, as a matter of fact, we're about to embark on that because we haven't really looked at what the immune system of kids whose mothers have these autoantibodies look like. That's something that one of my graduate students is going to work on now. But the other, and she just had a baby, so she's out doing her own own nurturing at the moment. But we will look at that. But the maternal immune activation model, when we've done it in non-human primates and mice, we do see an effect of maternal immune activation during pregnancy on how the immune system develops in the offspring. So we do see that there is an effect, and that is something that we're exploring in terms of what that means for future exposures. I think that's really important. On the other side, looking at how the environment affects the immune system of a child postnatally, while we don't necessarily understand how much is prenatal and postnatal in terms of exposures, what we do know is that some children with autism respond very differently when we expose their cells to certain toxicants like the brominated diphenyl ethers, which were in flame retardants. I mean, they've been pulled now, but they are long-lasting in the environment. And so that class of chemicals, for some reason, children with autism, when we take their cells and expose them to them, react very differently than they're typically developing controls. And it looks like it's due to a change in demethylation of the regulatory cells. So we're trying to work backwards and to understand why that is and how that relates to neurodevelopment. I really don't know yet. And so, I mean, those are the questions that we're working to answer.
Okay, and stepping even further back, what about perinatal exposure? So before they're pregnant, the things that happen, insults and influences prior to, like in the preconception period?
And that is a good question. And some of it is how long lasting are those effects? I mean, we know that prenatal, or preconception increase in folate is beneficial, right? That you have a good level of folate on board at conception. And so planning pregnancy, that's important. I think what we don't understand is and we're looking at, actually we're trying to examine, is body burden in the mother of any prenatal exposures, and because they're long lasting, they're going to be in the system for a while, throughout life quite often. And so how that affects the change in outcome, we actually are working on that. That's part of the MARBLE study that's going on here.
Okay, somebody's asking here about IVIG. So
IVIG.
Yeah, IVIG on animal models. And have there been any studies or tests that you're aware of that relate to the antibody research?
No, we haven't tried IVIG as a therapeutic in terms of the maternal autoantibodies yet. We first had to create the model so that we could do that, and it wasn't going to work if we did a passive transfer and then put a ton of IgG in there. So we had to create the model that we just made to be able to test that. But it is a potential therapeutic.
Okay. So the next question is about, they're asking about, and I don't know if this is anything that you've addressed in your research, so I'll just throw it out there. They're asking about maternal factors like MTHFR. They're also asking about Rh positive and negative and how those things can relate to the maternal fetal autoantibodies, like what the connections are with them.
We haven't found a connection with the MTHFR is really more with folate related, so we haven't found a connection with that at all with the autoantibodies. Not that we won't look further, but so far we haven't. The so sort of the other we also haven't found a relationship yet, but that also doesn't mean that there isn't one. It's just we haven't been able to. The MET gene is the one that we've found the best relationship with so far.
Okay, so that's your strongest signal. So that's
our strongest signal, yeah. And I think it was all but one mother, so had a polymorphism, which is a lot of mothers have the polymorphism and don't have a child with autism. So clearly, it's a case of susceptibility, and then another hit being needed to push it over the threshold.
Okay, this is an interesting question. This person was really listening to what you were saying, and she was talking about the different phenotypes. She said, as we sort of unravel these threads and we start to see the phenotypes evolving, what do you think about creating different immune-associated disorders that are sort of distinctive and discrete, rather than calling them all autism? Or do you see that they're all common enough that they would fall under the same umbrella? What do you anticipate, I think is what you...
No, I think autism as a diagnosis is based on behavior, right, at this point. I think subtyping autism, the benefit for doing that is to hopefully target therapeutic, to be able to target therapeutics and be more effective on that front. And so calling it a new disease, I mean, we've labeled, the maternal autoantibody associated autism, MAR autism, but only because it helps us distinguish that these kids potentially, this is their etiology, right? This is how they got to the disorder. I don't know that we'll change, I don't know that it would be good to change diagnosis completely, but I think if we could at some point get to the level where we have biomarkers that tell us which category they fall into in terms of causation, or what their pathway is going to look like, what their physiology is going to look like, we can be more effective in how we target treatments to those. And I think that's the biggest benefit of phenotyping in terms of understanding how they got there and preventing and or treating, postnatally.
Okay, we only have time for one more question. I know you need to go, but the question is there a commercial test available to test the mom for these maternal fetal autoantibodies?
Yeah, we've been working on that for a while. And I think one thing about science is, on paper, something seems very simple until you try to execute it. So while it should have been very easy to develop this test and get it commercialized, we actually ran into some issues with some non-specific staining to the proteins that we were using, because they have a tag on them to help you purify them, and all sorts of things that we're fixing. It's just taking me a lot longer than I thought it was going to. So we did form an entity here at the university to try to, through the university here, to try to make this available to families. And as soon as it is, I will let ARI know, because I will be very happy when we actually can help and make this useful and helpful.